Good afternoon, everybody. Welcome back. Hopefully, everyone had a good lunch break. So on behalf of my co-moderator, Dr. Huzeyfe, Yilmaz, who is a senior general physical scientist in OPQR, it's my pleasure to welcome everyone to the session 2, Regulatory Science Applied to Ophthalmic Dispersion Products. This session will build on the scientific and regulatory foundations that I laid out earlier today and turn our focuses towards more complex ophthalmic dispersions, including suspensions, ointments, and a little bit discussion on the emulsions as well. Our speakers will present coordinated perspectives from different focuses. We'll begin with Dr. Vatsala Naageshwaran, CEO of Franklin Biolabs, discussing topical and intracameral pharmacokinetics of complex ophthalmic suspensions. Although she's not able to join us today due to an unforeseen schedule change, she has kindly provided a recorded presentation for the session. Next, we'll hear from Romit, Jani, Senior Director of Formulation R&D at Solaris Pharma, will address development and optimization of particle size for ophthalmic dispersion formulations. We will then hear from Dr. Xiuling, professor at the University of Connecticut, presenting in vitro and in vivo assessment approaches for tobramycin and dexamethasone ophthalmic ointments. Following that, Dr. Chung, lead pharmacologist in the Office of Bioequivalence, OGD, will provide a comprehensive overview on bioequivalence perspective for common challenges and deficiencies. Then the session will transition into model-informed regulatory science. Dr. Jessica Spires from Simulations Plus will present on PD extrapolation using ocular PBPK modeling. Dr. Mingliang, Senior Pharmacokineticist in the Division of Quantitative Methods and Modeling in OGD, will present on PBPK PD approach for bronzolamide suspensions. And last but not least, Dr. Carrie German of CFD Research Cooperation will present on in silico modeling for ophthalmic suspensions. Collectively, these presentations will provide unique and complementary insights that hopefully will advance a robust regulatory science framework for the efficient development of complex generic ophthalmic dispersion products. So without further ado, we will proceed with the recorded presentation from Dr. Vatsala. I'll leave the podium to her recording.
Good afternoon, and thank you to the FDA and CRCG for inviting me to participate in this exciting workshop. My name is Vatsala Naageshwaran, and I will be presenting on the topical and intracameral pharmacokinetics of complex ophthalmic suspensions. To start with a little background, we're all aware that topical drug administration is widely used for treating ocular diseases because it can be used in outpatient settings, and topical applications are relatively easy. However, there are many challenges with topical drug delivery that involve factors that limit the drug absorption to the eye, such as tear turnover, nasolacrimal drainage, the tight corneal epithelium, and systemic absorption at the ocular surface. There are reports on the pharmacokinetics of topical eye drops. However, information on absolute drug bioavailability to the aqueous humor is limited, especially for topical suspensions. Therefore, preclinical studies can really provide a good understanding of the pharmacokinetics and ultimately the pharmacodynamics of such topically applied ophthalmic drug products and determine what is the impact of the critical quality attributes of the formulations. When combined with modeling, this information can be valuable in supporting the development of generic products and novel delivery systems with improved pharmacokinetic profiles that could benefit patients. Complex ophthalmic formulations like suspensions are needed in the delivery of poorly soluble drugs to the eye, an organ with multiple compartments and diseases of variable etiologies. Unlike other drug delivery routes, a topical ophthalmic formulation delivers the drug to the ocular tissues in a very short time frame. The instilled drug is eliminated rapidly from the ocular surface, and only a small fraction, less than 5% of the instilled drug, reaches the inner ocular compartments. When you have formulations with different physicochemical properties, they can have significantly different PK and thereby in vivo performance. But as mentioned, the impact of these differences has not been systematically evaluated. Rabbits are a very good model for these ocular PK studies, since the rabbit's eye shows many anatomical and physiological similarities to the human eye. We'll start by looking at the absorption and elimination pathways following topical and intracameral administrations to set the stage for understanding these PK studies. So in the schematics, we see the absorption, distribution, and elimination pathways following topical administration and intracameral administration. With topical administration, absorption has to take place across the cornea or conjunctiva sclera. Elimination can occur at the ocular surface from the tear fluid through tear turnover, nasolacrimal drainage, systemic absorption via the conjunctiva, and from the aqueous humor through the outflow, which is via the trabecular meshwork and the uveoscleral outflow, as well as the blood flow of the iris ciliary body. With intracameral administration, the drug is deposited directly into the anterior chamber, similar to an intravenous administration, and it represents 100% of the drug in the aqueous humor, from where it can be eliminated by aqueous humor outflow via the trabecular meshwork and uveoscleral outflow, or the blood flow of the iris ciliary body. In both instances, distribution of the drug occurs in all the major compartments, such as the aqueous humor, iris ciliary body, lens, and so on. Now, vision loss in glaucoma is related to elevated intraocular pressure, which can injure the optic nerve and eventually lead to blindness. IOP is determined by the dynamic equilibrium between the production of aqueous humor in the ciliary body and its outflow through the trabecular meshwork and Schlem's canal in humans and through the intrascleral venous plexus in rabbits. Aqueous humor production can be reduced by the usage of both topical and systemic carbonic anhydrase inhibitors. In 1998, a topical carbonic anhydrase inhibitor, brinzolamide, was approved for clinical use. Commercial product is called Azopt. This is 1% brinzolamide ophthalmic suspension formulated at a pH equivalent to that of human tears and thereby designed to be more comfortable to enhance patient compliance. The chemical structure and some physicochemical characteristics of brinzolamide are presented in the figure. The pK values of brinzolamide allow it to act as an acid or base depending on the pH, and at a pH of 7, brinzolamide has low water solubility, which results in it being a suspension at neutral pH. This is a description of the ocular PK study that was performed in rabbits to determine the comprehensive ocular pharmacokinetics of brinzolamide in relevant ocular and systemic compartments of rabbits. New Zealand white rabbits, or so-called albino rabbits, were dosed with via topical, intracameral, and intravenous administration and various ocular compartments such as tears, aqueous humor, iris ciliary body, as well as systemic compartments such as whole blood, processed to plasma were analyzed. LC-MS methods in these matrices were developed for the analysis of brinzolamide, and they were qualified. The data was then explored by non-compartmental analysis using Phoenix Win non-lin, and the absolute aqueous humor bioavailability of brinzolamide was calculated and reported for the first time. After intracameral administration of fully solubilized brinzolamide, the aqueous humor concentration curve was well described with a two-compartment model. Brinzolamide concentration in iris ciliary body declined slowly only after 8 hours, and the concentration ratio between iris ciliary body and aqueous humor was much larger at the last sampling point compared to the earlier time points. After topical administration of 1% brinzolamide suspension, the brinzolamide concentration in iris ciliary body was consistently higher at all time points compared to the concentration of the aqueous humor. Brinzolamide PK parameters calculated by non-compartmental analysis are captured in the table. Brinzolamide is not known to undergo any metabolism in the eye, and some primary metabolites have been identified only in blood and feces. Therefore, aqueous humor turnover and drug elimination to iris ciliary body blood flow are the only possible routes for drug clearance from the anterior chamber. So the intracameral clearance of brinzolamide was found to be about 4.12 microliters per minute, which is only slightly higher than the normal aqueous humor turnover of about 3 microliters per minute in rabbits, which suggests that the elimination to the iris ciliary body blood flow is a minor contributor. The high volume of distribution of brinzolamide after intracameral administration is greater than the volume of the aqueous humor, which is about 250 microliters in rabbits, and this indicates high drug binding to the surrounding tissue such as iris and ciliary body. Thus, the affinity of brinzolamide to the carbonic anhydrase enzyme, which is present in the ciliary epithelial cells, seems apparent. After intravenous administration of brinzolamide solution, with a 2-week sampling period, the elimination half-life in plasma and whole blood appeared to be higher than 2 weeks, as can be seen in the figure. Therefore, the PK parameters could not be estimated. Brinzolamide concentration in whole blood was also much higher than in plasma, indicating drug accumulation into red blood cells. After 8 hours, whole blood plasma and concentration ratio remains constant. And at the terminal time point of 14 days, the concentration in iris ciliary body and aqueous humor were measured. Aqueous humor was below the limit of quantitation. However, iris ciliary body concentrations could be reported. This long elimination half-life seen from the present rabbit study was consistent with human data that's been published in the prescribing information for AZOP that indicates that after topical administration, brinzolamide is absorbed into the systemic circulation and distributes extensively into the red blood cells, exhibiting a long half-life in whole blood. The concentration of brinzolamide in aqueous humor, iris ciliary body, whole blood, and plasma at the last sampling time points after the different administrations and the corresponding ratios with respect to the iris ciliary body are reported in this table. Brinzolamide concentrations in the iris ciliary body were much higher than the levels in the aqueous humor at the last time points, both after topical and intracameral administrations. The iris ciliary body aqueous humor ratio at 12 hours after topical administration is much higher than the ratio obtained following intracameral administration at 24 hours. In fact, the ratio obtained from the extrapolated topical 24-hour data is even higher. Similarly, the ratio obtained following intravenous administration was also high. In conclusion, the absolute bioavailability of brinzolamide in aqueous humor was determined in this study and calculated to be 0.1%, which is 15 to 45 times lower than the bioavailability of solutions like timolol and betaxolol. The ratio of the dose-normalized AUCs in iris ciliary body and aqueous humor after topical administration was greater than the ratio after intracameral administration. And this suggests that the major part of brinzolamide in the iris ciliary body does not enter the tissue via aqueous humor. The brinzolamide concentrations were higher in the iris ciliary body than in the aqueous humor, even at early time points following topical administration, further supporting the role of direct drug absorption across the conjunctiva and sclera to the iris ciliary body. Two factors are thought to contribute to the non-corneal absorption. Dissolution of the suspended drug particles may take place in the conjunctival sac, being in direct contact with the conjunctiva, And the ex vivo permeability of brinzolamide in rabbit and porcine conjunctiva has been shown to be an order of magnitude greater than in the cornea of the same species. Moving on to another ophthalmic suspension, which is the corticosteroid class of drugs. Topical ophthalmic corticosteroids such as dexamethasone have been used for over 60 years, and the clinical benefits in managing inflammation have been very well established. Like many poorly water-soluble corticosteroids, ocular dexamethasone products are suspensions. There are multiple dexamethasone suspensions on the market, including Maxidex, Tobradex, and Tobradex ST, which is a combination of dexamethasone and tobramycin. An ocular PK study was similarly performed in rabbits to determine the pharmacokinetics of dexamethasone in various ocular matrices following topical administration of 3 commercial dexamethasone products, Maxidex, Tobradex, and Tobradex ST. Intracameral injection of soluble dexamethasone was given to obtain the kinetic parameters with non-compartmental analysis enabling quantitation of dexamethasone bioavailability. Bioanalytical methods using mass spec were once again developed in various New Zealand white rabbit tissues, ocular tissues, and the data was explored by non-compartmental analysis using Phoenix Win-Non-Lin, and the absolute aqueous humor bioavailability of dexamethasone was calculated and reported for the first time, as well as compared between the 3 commercial products. After intracameral injection, the highest concentration of dexamethasone was seen in the cornea. The concentrations in the iris ciliary body and aqueous humor were similar, and the concentrations in the conjunctiva were much lower. Accordingly, the highest Cmax and AUC were obtained in the cornea, followed by iris ciliary body, aqueous humor, and conjunctiva. Since the measured concentration profile in the conjunctiva could not be determined beyond 2 half-lives, the terminal half-life and AUC infinity were not determined for this compartment. The primary PK parameters were estimated using aqueous humor concentrations. Clearance was determined to be 13.6 microliters per minute, and the volume of distribution at steady state was 990 microliters. The mean concentration time profiles in 3 compartments, tears, aqueous humor, and cornea, were compared for the 3 topical formulations, Tobradex, Tobradex ST, and Maxidex. The pharmacokinetic parameters after topical administration for the 3 products were determined by NCA as shown in this table. And as expected, dexamethasone concentrations were in all cases highest in the tear fluid, followed by cornea and aqueous humor. What we observed was that the AUC values of Tobradex ST in cornea and aqueous humor were higher than the values of Maxidex at an equal topical dose of dexamethasone. Tobradex also resulted in having higher AUC values than Maxidex, but 2 times a higher dose of dexamethasone was administered in Tobradex compared to Maxidex. The Cmax for tear fluid was determined for all 3 products as well as the Tmax. However, due to rapid loss in the tears following topical administration, only partial AUCs were determined for an early duration of 0 to 10 minutes when the majority of drug elimination takes place in tears to provide a more accurate picture. And the AUC 0 to 10 minutes for this time period is reported in the table for the 3 products. The tear film kinetics of these different formulations were studied by comparing that partial AUC of 0 to 10 minutes. Dexamethasone exposure in rabbit tear fluid was highest after instillation of Tobradex ST as compared to Tobradex and Maxidex. Tobradex ST, as you may be aware, includes gel and gum that prolongs its retention on the ocular surface. This matches results that have been previously reported of much higher dexamethasone and tobramycin concentrations in the tear film following administration of Tobradex ST compared to Tobradex. In accordance with the tear fluid results, Tobradex ST delivered similar concentrations of dexamethasone to the cornea compared to Tobradex, even though the drug dose in Tobradex ST is half that of Tobradex. The absolute bioavailability of dexamethasone in aqueous humor was calculated based on the dose normalized AUC infinity in aqueous humor after topical and intracameral administrations. While Tobradex is effective and widely prescribed, the newer formulation, Tobradex ST with 50% lower concentration of dexamethasone, but containing the viscolizer xanthan gum, was proven to improve the ocular bioavailability of dexamethasone. In vitro studies showed that xanthan gum in Tobradex ST creates a different shear thinning and higher viscosity than Tobradex with hydroxyethyl cellulose. Upon dilution in tear fluid, the viscosity of Tobradex ST increases over 5-fold, while the viscosity of Tobradex decreases by half. Head-to-head clinical trials have shown that dexamethasone concentration in aqueous humor was the same or even higher following instillation of Tobradex ST compared to Tobradex, making them clinically equivalent, even though the dex concentration in Tobradex ST is only 0.05% compared to 0.1% in Tobradex. Thus, the results from the rabbit study were very useful in providing the critical pharmacokinetic parameters of dexamethasone, including bioavailability and clearance, which correlates well with the published formulation characteristics as well as the clinical data for these products. Identifying critical quality attributes for complex local acting ophthalmic formulations and establishing in vitro in vivo correlation can facilitate the selection of appropriate thresholds for formulation changes that reflect lack of impact on in vivo performance. FDA-sponsored research under an IDIQ task order to identify the potential impact of critical quality attributes by testing azopt and 5 other brinzolamide formulations that varied in particle size distribution and viscosities through topical studies in rabbits. The ocular pharmacokinetics was determined in multiple ocular tissues for the different formulations, and these results were then utilized to determine the between and within animal variability, and the variances were then used to run a power analysis to determine the sample size for a different study that identified some benefits of a paired eye study design. I'm not going into all the details in this presentation today, since this is out of scope, but the paired eye design that was identified could be valuable in determining the IV-IVC between formulation changes and PK parameters and help to determine the tolerance thresholds for inactive ingredients. Modeling the data from such studies could help to establish the thresholds for changes in generic formulations that are being developed for critical IOP lowering ophthalmic products like brinzolamide. I would like to acknowledge and thank Professor Arto Urti, Lecturer Belipeka Ranta, Docent Eva Del Amo, Dr. Glenwood Gum, Biostudy Solutions, and the funding that was made possible by a US FDA contract that supported these studies. Thank you very much.
Once again, really appreciate Vatsala's effort on making the recorded presentation despite her schedule change. Next, we will have Romit, Jani from Solaris to present on the development and optimization for particle size of suspension formulations.
Hello, everyone. Thank you for providing me the opportunity to discuss some of the important topics for ophthalmic dispersions. I think this is a great forum where industry, academia, and FDA meets and discuss some of the challenges that we are facing for development of complex products. So today, I think we have already discussed a little bit about particle size and importance of particle size for bioequivalence, because some of the complex ophthalmic products have very challenging release methods. And because of that, I think this topic is a little bit more important. How do we develop those methods? So today in my presentation, I am going to provide two different case studies where we are trying to develop particle size method for ophthalmic suspension as well as ophthalmic ointments. Of course, particle size impacts bioavailability, ocular irritation, and physical stability, but now even it is more important, as I mentioned, that some of the product has complex release methods, and particle size method play a great role, showing that how we can show the bioequivalence for these products. So the first example I have is that aqueous-based suspension products. For this particular product, these are some of the criteria or stepwise optimization that we have performed by selecting right dispersant, equilibration time, stirring speed, sample loading, and obscuration range. The first one is, of course, the selection of dispersant. It is important. This particular product was aqueous based, and API is practically insoluble, lipophilic in nature. Water was main ingredient in formulation, so we have selected water as one of the dispersant for developing a method. In terms of obscuration range, it is little interesting that signal to noise ratio is very critical in this case. If you see right hand side at bottom, at the low obscuration range, we don't see a good signal to noise ratio. However, for all obscuration ranges, we have seen a pretty good particle size distribution, but because of very low signal to noise ratio, I think we need to exclude the lower obscuration ranges. We have challenged these obscuration range 3 to 5%, 5 to 10%, and 10 to 15%. In the lab, we have performed multiple run and we have got very tight results. There were no ghost submicron peaks that was observed even for all three obscuration ranges, and high enough signal to noise ratio was seen. So for this particular method, we have selected obscuration range accordingly. Another parameter was ultrasonication. Interestingly, for this particular product, we have challenged different ultrasonication at different timings, and also we have evaluated no ultrasonication. If you can see the graphs, the one in the red is without sonication, and the other two are with 50% sonication for 3 and 10 minutes. We have clearly seen that sonication at 50% for that many time is not helping in terms of getting the right distribution. So we have excluded those particular sonication percentage. We have also challenged 10% sonication at the lower sonication with 3 minutes as well, and the results are not different. So for this particular product, we have selected no ultrasonication for the development of a particle size measurement method. The next slide, we have equilibration time. We have also challenged those because it is important to prevent transient agglomeration or sedimentation. the particle size results obtained at different time of equilibration. If you can see 10, 20, and 30 minutes, the particle size results obtained are not a lot of difference between those, and so for this particular method we have selected the time 10 minutes. Stirring speed is also one of the important criteria that we are challenging for development of the particle size distribution method. Of course, the product has some surfactants, so higher speed creates some globules and give us false results. We have challenged 500 RPM to 2000 RPM stirring speeds for to see whether or not are we getting any different distributions. And with all the three challenge parameters, we are getting a good results, and so we have selected correct stirring speed of 500 RPM. Sample loading or dilution, this is also interesting. One of the thoughts that came into our mind, that whether or not do we have to dilute our product every time in order to get good distribution, but the answer is for this particular product is no. We have challenged the product as is versus 10 and 20% dilution with water, and the results are pretty same for all the three different dilutions, 2 different dilution and without dilution. So without getting into a manual intervention and of dilution, we wanted to avoid those, and we selected no dilution for these. So based on these, I think we have finalized our parameters for suspension product, and this is the compilation and justification for that what I have discussed. Interestingly, the next slide is for the ointment-based particle size measurement. This is little tricky. This is not something that is very easy to get hold on particle size. We have tried different approaches. We are still trying to get the final method, but so far whatever we have performed, I am going to explain in this presentation. So for ophthalmic ointment, of course, to develop those methods are tricky because particles are embedded in lipophilic base. Measurement of particles from an ointment is very tricky. We have used microscopy method with image provision camera and software for this particular product. And we have tried to compare the dry API. We have compared the placebo as a baseline, and then we have compared innovator and test product. Of course, this particular method is still in development phase, but phase 1, what we have generated data, I wanted to show today. As a dry API, we have seen these numerical distribution, and we have applied some of the filters on these and get the particle size distribution of D10, 50, 90, and span on a dry API. So this was the first step where we got an idea about particle size distribution of an API. And one of the interesting aspect of this particular software also is that we can select each and every particle, and we can get idea of morphological values as shown here. Circularity, which shows how smooth or round particles are. Aspect ratios, whether or not they are in rod shape, needle shape, or crystals. Solidity, which gives us an idea about how rough the particle surfaces are, and of course span data. So based on these, we get an idea that for each and every particle we select, what are the different morphological values. And this is, I think, one of the important information that we are getting by these. We have evaluated test product as well. We have generated some similar data on test products with numerical distribution, and of course with some filters trying to nullify placebo impact artifacts and some aggregates. we have got those values of 10, 50, and 90. And for this particular test product also, we generated all the morphological values. With reference product, similarly, we have performed the same experiment and generated some values of D50, D90. And this is particular technique has a lot of challenges because the data that we have generated is as is without disturbing the ointment. And that is one of the discussion point, whether or not how do we want to not disturb the ointment and we get the accurate results for ointment products. We are going to challenge this particular method with also with some kind of mineral oil dilution. We will also think of getting some solvent-based dilution on the ointment basis, but that work still to be done. We'll compare these all different methods and see which one works best for this product. As is measurement is tricky in terms of how do we apply the product on a slide, whether or not that is even and small enough that you don't get multiple overlaps on particles. So these are some of the challenges we are still evaluating. And this initial data comparison on a test and reference product, the method is still under development stage. And this is an example of one particular API dispersed in an ointment base. Things are getting even challenging when we have two different API in an ointment base, and the question arises that whether or not, how do we develop those methods? What are the important aspect or technologies, solid state NMR, Raman, or any other technologies we can use to identify each particles, whether it's API A or API B, and then of course get the particle size distribution on those. With that particle size, I wanted to go over with couple of formulation development challenges also, which was not a part of my initial discussion, but I have kept a couple of them since it is aligned with our topic. So with that, of course, sterile manufacturing is always a challenge. Whether or not do we have the right infrastructure for sterilization is always a discussion. One particular product that we have found the facility who can make products sterile by autoclave. Product had some impurities, oxidation sensitive impurities, and we have seen that pH and assay values shifted, as well as impurity levels are also shifted. So these are the challenges we have evaluated and try to find out what kind of sterilization technique we opt, and then we have changed the sterilization technique from autoclave to sterile filtration. And also we have added a nitrogen overlay and with some mixing speeds adjustment to get as much less air as possible, and we have seen the results were quite improved with 40° 6-month data. So for sterile dosage form, I think it is also very important which sterilization technique do we select, whether or not do we have the right infrastructure for those kind of techniques such as heat sterilization or sterile filtration. Another example is also, if you can see on the left, the product with a lot of air, and on the right we have optimized some of the manufacturing techniques to get as much layer as possible. So air entrapment issues, if you can see, it can create a lot of problem in fill weight adjustments. Of course, non-uniformity on the drop size, potential oxidation, and inconsistent product densities. So with all these problems, we have thought through and come up with process changes. And when we talk about process changes, it's in a non-sterile environment, it is a little bit easier to get those changes done. But when we talk about sterile environment, we have to be very careful that how can we resolve these issues and whether or not it has any potential adverse impact on the product or sterility. So we have performed some vacuum deaeration on a non-sterile phase, We have then sterile filtered the product to get rid of some of the larger air bubbles. In the filling side, we have introduced the bottom of filling, so the nozzle goes all the way in the bottom of the container and then goes up. In that way, we reduce a lot of air formation during the manufacturing. And of course, with some controlled mixing and vacuum applications, we have improved these air entrapment issues. With that, I think I wanted to thank everybody, and we will discuss a little more on a panel discussion specifically about particle size, distribution, and IVRT challenges. Thank you.
Thank you for the insightful presentation. Next, let's welcome Dr. Xiuling Lu from the University of Connecticut.
Great. Thank you for the kind introduction. I appreciate this opportunity to share some of our work related to ophthalmic ointment evaluations. In the morning, we have a great discussions about the challenges of all kinds of ophthalmic products. We all know that drug content is very low for these type of products. And also the drug release for ointments, it's pretty much from the surface layer. So the surface area will highly affect the release properties. And we still don't have really compendial method for IVIT tests and even understanding the impact of drug properties, for example, hydrophilicity, still as a question, what is the impact of hydrophobic, hydrophilic drug molecules and this release performance tests. And the most important, we kept asking ourselves, how is the relevant to other in vitro test to the in vivo result? So with all these questions in mind, we discussed and did quite a bit of study around this topic. We all know that in vitro release test, you can use standard like USP iris 1 or 2, or with the immersion cell, and you can use friend deficient cell. You can use USP iris 4 with the semi-solid adapters. Well, with all the methods we've been seeing, we would like to use the Toberdex, which is ointment formulation containing both hydrophilic drug, tobramycin, and hydrophobic drug, dexamethasone. And then we conduct in vitro and ex vivo as well as in vivo tests for these evaluations. From quite a few years of work, because due to time, we are not able to go deeper to what we had found in the past, but we understood that the ophthalmic ointment base, which is petrolatum, rheological profiles will highly affect the properties of drug release. We also understand that the crystallinity, for example, amorphous or crystal drug, will have different release performances. Typically, amorphous drug has high release for the profiles we observed. With all these considerations, we also compared USP iris 2 versus 4. We found actually iris 4, even though they can show more release, but they're more variable. So that's what we decided is to use USP apparatus 2 for our further studies. Well, and we all know that detection limit is a challenge, so that in order to make a surface area well-defined but larger, what we did was develop this adapter to have a two-side release. So that means if you look at this panel over there, we have a bottom part on the left side of this part. This is the bottom, and we can put a sheet of membrane on top of that, and then put spacer on, and you put ointment there, and put another sheet of membrane, and screw the last piece. So that's a good, well-sealed system that can set it up, mounted to the USP apparatus 1 or 2. Basically, we make it that self-rotating. So that is very easy to use, and also we are able to get very reproducible results. And we also compare, before we decide how to use or what setup we use, we also compare the different of the, like either you do one side versus two sides. If the surface area will affect the release performance, actually this is proportional, so the double of the surface area will not affect the release properties. And also we look at if you put the adapter on the bottom part, or you do apparatus 4, but the flow is going the bottom up. So all these comparisons, we have paper about the differences of the impact of the adapter setup on the release properties. Well, essentially we define that this release setup is the best for differentiating our drug formulations. And the next, we actually wanted to expand a bit further. Okay, we do in vitro release, but how to correlate that or understand the differences between in vitro and in vivo? We want a little intermediate segment. So that's what we did, is do a ex vivo test. What's the difference between in vitro, ex vivo? In the sense of, yes, we have a release compartment, and for the ex vivo, sorry, if I go back one more, if you see that a little adapter, which has very small release media on the bottom, so put a rabbit eye on the top of this holder, and the front side of the eyes only touch the release media, so that we are able to get a small volume release and with a biological subject, that's the rabbit eye. The difference between ex vivo and in vivo is we don't have drainage. So basically there's no flush, and you have a steady of the release over there. And so then we also want to look at the in vivo performance. That's our study, to look at in vitro, ex vivo, and in vivo results of the ToberDex, which has 2 drugs. And we also compared formulations, as Q1, Q2 formulations, with the ointment from different vendors. They have different rheological properties. One thing for a release test that we compare, you often case when you have hydrophobic drug, you use surfactant. So 280 is often used. Then we wondered, using or not using 280, what is the effect of the surfactant use in the release property? So the ex vivo, we do not have a surfactant, because we know for the biological subject that you will not have surfactant in there. And we want to compare from in vitro test. We want to discriminate all the formulations and then go to see what's the correlation between in vitro and ex vivo of that, more towards relevant to biological environment, and then go down to in vivo. So the next is to do the in vivo test. Actually, this collaboration with the VCU, Dr. Qingguo Xu is in one of the audience. And we did the rabbit test with 3 rabbits per group, but 6 eyes per formulation. And we have various time points. Each time points determination, we took the tear film and all the eye compartments and did an analysis. So the first thing we want to see, all the formulations we prepared, even though they're Q1 and Q2, but you see the rheological profiles, they're very different. For ToberDex, which is blue color, And then we have one group with the green color that has lower viscosity in general, and then we have another group with spectrum has higher viscosity in general. We have one with very close, as IGI 386 is very close to Tobradex. But you could also see a slight difference in terms of the lower temperature viscosity is slightly higher, higher temperature viscosity slightly lower. So there is some distinctions. Okay, so the next is to compare two different drugs. We want to just go one by one of the two drugs. The first is dexamethasone, which is relatively hydrophobic, and it's quite busy, but the top panel data set is the release without twin 80, and the bottom panel is release with twin 80. And we have release profile with the release amount versus time plot. And as Dr. Xiaoming Xu already mentioned earlier, there are different ways of plot the time scale. You could go with a regular linear time scale, or you can go with a log time, or a Gucci model of a square root time model. So we plot all three of them, and then in order to understand the release rate, we can see, yes, you have different release amount. Amount could be one consideration, but when we consider, amount of drug at the given time, which is a release rate. So rate is more important than the amount at a starting point. So we look into the release rate differentiation for the ones with and without twin 80. A lot of data, but if you look at the blue bar, which is Tobradex, and it shows all this, significance compared with the different other formulations, regardless what way you plot the time scale, meaning that the release rate are different in terms of the formulations. When we talk about the formulation without twin 80, remember this is hydrophobic drug. And when we have a release media with twin 80, that's commonly used because we want to enhance the release in order to get, for example, detection and faster dissolution. So, but with twin 80 for this hydrophobic drug, you're not able to discriminate that well compared to the formulations across, not as well as the formulation without twin 80. So the release media without twin 80, you're not disturbing the drug dissolution. You're not affecting the dissolution. That's more towards the innate dissolution properties of the hydrophobic drug from the ointment. So on the right part is the R squared. You look at it, that it's quite linear for the release rate determination. Okay, so that was just the in vitro test, and now is ex vivo release test. For ex vivo, we looked at the release in the little compartment, which has very small volume. It's just 670 microliters. And we also took the aqueous humor as well as cornea. So those are the critical components that, at least for hydrophobic drugs, we expect that they are able to cross cornea to go to aqueous humor. So in terms of the release media, you can see that I can go to the next because we look at the release, more a discrimination, that all is different and similarly to the in vitro test. And for the aqueous humor and the cornea, you're also able to see the differences between these formulations. Only the aqueous humor is more, representing the difference between formulations, very consistent with the in vitro result. Well, the next is in vivo. So first I have to acknowledge that it's not an easy task, and we do tier film, and there you can see the data plot, and there's a standard deviation very, very high, all over the places. So it's a challenging for do a practical collection of tiers, and we've been seeing the variations of our samples. And it's interesting to see that the cornea dexamethasone still show TOBR dex is the highest, very consistent with ex vivo and in vitro test, but not much of an aqueous humor. So it's probably the first step is get to cornea and then going to the aqueous humor. The time scale is not reaching that distinction. Well, and then if you look at the tiers, as I mentioned, the variation is very high, and pretty much there's not much distinction we can tell for, looking at the tiers. If we use stats, looks like IGI 386 has significance difference compared to TOBR dex. And we're trying to understand what happened to this. It looks like the release performance is, for TOBR dex is supposed to be better, but why the tiers, besides we see the variation, they see like a first time point, the red color showing on the spot, that's the first time point, and that is very high. And just early time point variations probably contribute largely to this. And one more thing probably I'll note after this is we have to be mindful about in vitro ex vivo, the distinction compared to in vivo, because there is a drainage and there is a retention time. And for ointment, retention time is higher. So that could contribute if you have a relatively higher viscosity formulation, then the retention could be more, and compared to lower viscosity, that could contribute to more of the tier. Right. So, well, this is quite a lot of numbers showing here. We just highlighted red in terms of the significant differences. We can see the tiers showing differences, as well as cornea drug concentration for Cmax and EUC shows differences. Overall, we want to kind of summarize. It's hard to conclude and make a very solid conclusion because we want to summarize what we learned. And in terms of in vitro release tests, if we are targeting discriminating formulations, I think for the release media, you do not add a 280 is more discriminating, the most sensitive one for the hydrophobic drug. And yeah, you're adding 280 is still discriminating quite good. But for ex vivo, it's relatively sensitive and relatively consistent with the in vitro tests. For in vivo, it's highly variable, and I think cornea slightly is kind of consistent compared to all across the formulations. That's what we can get from the data of hydrophobic drug. And then come to the hydrophilic drug, it's very interesting. Think about hydrophilic, it's very highly soluble, water soluble, and instant dissolved in aqueous. So in terms of the formulations, you kind of can see a slight difference for the group versus the one on the bottom, which has the highest viscosity for this one. It looks like a distinction, but I want to show you again for release rate. We want to look at release rate. And in terms of release rates, we are only able to get the significant differences for the formulation, for the dissolution test with 280. You have to have a surfactant there, which is interesting. Why you have to have surfactant to be able to discriminate all the formulations? So what we feel is the surfactant actually is kind of like interact a little bit with your ointment base. So that would more or less because of the interaction with the ointment, it could affect or get a more release and then distinguish the ointment-based formulation differences. So if you just think about all the surface release hydrophilic drugs is instantly released, you wouldn't be able to see the differences. Just like how you try to manipulate a little bit. Now we did change the some performance of the base, and then we can get more release, and then we can see the differences between formulations. So for ex vivo, there's no surfactant over there. And then it's pretty much for the release, the first figure, you can see that the release is no distinction between all the formulations. And the next one showing the release rate, and there's no significant difference for any of those formulations. It doesn't matter if it's in release media or aqueous humor or cornea, which is kind of understood. Okay, so the next is for in vivo study. Again, there's a variation. It is highly variable as well. And then thinking about your drug is so water soluble, and that variation could be even higher compared to you think about the hydrophobic drugs. So you see the first spot over here again, that red is IGI 386, and that is very high for the early time point release. And if I just do not consider that time point, and then if I just make the cut for go by 5 minutes later, bypass of that time point, and then other formulations, not that very different. So just to keep in mind of that. And one thing we saw very interesting, we do not expect aqueous humor tobramycin content. So this is a hydrophilic drug. It's really, very not expected to go cross cornea. And now here we do see quite a bit of the drug from tobramycin and tobradex. Sorry, the tobramycin and tobradex actually got a higher drug content in aqueous humor. Well, we could put some speculation there. I'm not sure this is a safe assumption, but there could be a few reasons. For example, the first, again, we also see high-tier, high-tier drug content for two formulations, which has a relatively higher viscosity. But if we say that the tiers will not be representing that well of the real drug release, because your release is very instant, you still have quite a bit of release in the environment and then drained out. And the other thing is, if you look at let me see, go back to the cornea and tobramycin, just the aqueous humor, why aqueous humor has very high drug content. We were thinking maybe there is some interactions affect the permeability of tobramycin. That's the only thing right now we can speculate a bit. It's a true test. I think we validated. I hope our collaborators already, tried a few times and confirm that result. It's true in the aqueous humor. So we hope it's somewhat is related to the formulations or the some component or even ointment affect the permeability of a cornea. Okay, so, and that's, I want to go just directly to the conclusion, trying to make some conclusive statement. what we can say is for very hydrophilic drug, tobramycin, if you want to discriminate the ointment formulations, you could use surfactant then that manipulate the ointment a bit so that we are able to discriminate the products better. And for ex vivo, we really do not see any differences across the formulations. But when you get to in vivo with a complex situation, we have to be mindful of the retention of the ointment and the potential effect of the drainage, and I'm not sure we can put that into conclusion why the core aqueous humor has a very some amount of a hydrophilic drug in there. So but we were thinking like in the later we could do more study looking into the permeability and influences of the formulations. That's the one consideration. With that, because limited time, we will do to cover a lot, but we have a series of publications in past years about all the in vitro tests and the different methodology development, membrane effect, and trying to correlate in vitro, ex vivo, and in vivo. Really appreciate the support from FDA and our collaborators that are wanting and Dr. Andre Barons and to Dr. Xu and the audience and all VCU folks and my lab members. Thank you so much for your attention.
Thank you, Dr. Lu, for the interesting and comprehensive technical report. Next, let's welcome Dr. Hee-Sung Chung from the Office of Bioequivalence to talk about common deficiencies in ANDA reviews.
Thank you to CRCG organizers for opportunity to present today. As mentioned, my presentation will focus on bioequivalence deficiencies related to complex ophthalmic drug products. This is my disclaimer. In my presentation, I'll go over what are the complex ophthalmic drug products, typical PSG recommendations for these drug products, followed by the general overview of the bioequivalence deficiencies that the agency has issued over the past 3 years, and conclude with the key takeaways and the acknowledgement. So GDUFA 3 commitment letter defines a complex drug product. And as you might see, maybe like 2/3 way down, there's a complex ophthalmic drug products. And so for the purpose of today's discussion, ophthalmic solutions will be considered simple drug products, while gels, emulsions, and ointments and such should be considered complex drug products. Going later in the day kind of helps because some of the topics were already covered by the previous presenters. Dr. Bruce Lerman went over extensively on challenges related to the bioequivalence demonstration for the ophthalmic products. So I won't go over that in too much detail. One way to mitigate these challenges in vivo studies might be to use an in vitro approach, but I think we have heard today that even with that, there are many challenges related to formulation as well as developing the in vitro methods. Interestingly, Andre and I are using the same PSGs to showcase the PSG recommendation. So I won't go into too much detail here, but one key item I would like to note is that in order to use the in vitro bioequivalence approach, Q-on-Q sameness is a requirement. And depending on the dosage form, for the in vitro approach, you may see recommendations for PSD study, while for others, you may see a global size distribution study instead. You see IBRT study recommendation, also the aqueous humor study. I note here that all the ANDAs that I've looked into from the past 3 years for the complex ophthalmic products, none of the ANDAs contained aqueous humor study. So let's take a look at the bioequivalence deficiencies. To provide a little bit information on the data set that I looked into, with the help of the project managers from our division, we pulled the original ANDAs from 2022 to 2025 up to August 25. And then among these products, we pulled complex ophthalmic drug products, and that landed us with 29 ANDAs for suspensions, gels, and emulsions. Among these, 21% contained GSD studies, 79% contained PSD study, and since none of these applications had aqueous humor study, and then they contained IVRT studies. Deficiencies were from the original cycle, so for example, DRLs and CRLs. And for the purpose of today's talk, I left out the deficiencies issued related to the studies that are no longer recommended in these PSGs. And you note the acronyms are spelled at the bottom of the slides if you need to see them. Formulation was extensively discussed by Dr. Lerman and Dr. Harigaya this morning, so I won't go into that too much. When we see certain issues with the formulation information, we may ask for a clarification from you. One is if we see discrepancy within the components and composition table. So for example, if you have a person weight per volume versus person weight volume for volume, and after conversion, if those numbers don't line up, then we might see clarification to get a proper number for Q1, Q2 assessment. Sometimes we see incorrect amount listed for excipient. So for example, if certain excipient is to be converted in amount using a range of density rather than a fixed value, then we would expect the resulting number to be also in a range rather than a fixed value. It's possible that your formulation is Q2 the same as a reference listed product, but when the change is on the plus side, then we would need to justify the excipient level based on the agency-approved drug products. Additional item I'm to note here is that time to time, we see discrepancy between the components and composition table and the batch formula. And in those cases, then we're coordinating with OPQ to issue seeking clarification about what is the correct number. From bioequivalence perspective, we will use the components and composition table at face value for the Q1, Q2 assessment. So now let's go into the in vitro BSB studies. This is a high-level overview of the deficiencies that we have observed. First thing that came to me, I noticed was there are many, many deficiencies for these drug products compared to, let's say, ANDA for tablets. It wasn't uncommon to see more than 10 deficiencies in a CRL. So that was surprising, and it took a while to collect all the information. The other observation was that oftentimes these deficiencies were about missing information or incomplete information, which means we were not able to put together all the pieces to understand how the method was developed, how validated, and how you conducted the pivotal study. I'm not downplaying the complexities with the formulation and all the in vitro studies, but from the review perspective, if more complete submission can be provided, then I think it will help expedite the review process and then hopefully then reduce the review cycle for these applications. So for example, if you look at the first row of the table, oftentimes the study protocol was missing or the SOP effective at the time of the study was missing, or in some instances, the study report was also missing. And so having complete information in that regard will help us. And this upon not just in certain part of the drug development, but across method development, validation, as well as the pivotal studies. So consider a complete summation of all these documents. I go to the last line in the table. Sometimes study information was insufficient. So for example, study site information wasn't provided, and we would need that information for the inspectional aspect. And then in terms of the drug product used for these studies, sometimes COAs were not provided. If the modified test product was used for discriminatory ability of the IBRT study, then what changes were made to those past products would be very crucial in understanding relevance of those studies and results. So consider these aspects in your submission. Next several slides go into more details about the missing information or incomplete information. So I don't plan on going over all the elements, but maybe you can consider them as like a checklist before you submit the ANDA or during your drug product development to consider, do we have this or do we have that information in our submission? Do we submit this information? So you can consider them as a reference point. So let's start with the global size distribution study. You will see a lot of common themes across these slides. So starting with the method development, how the method was optimized and why you're using certain instrument parameters will be helpful in understanding whether method is proper or not. If you're not diluting the sample, then which refractive index you're using for that experiment also be helpful. But for GSC studies, oftentimes you want to dilute the samples. One of the things that we learned early on for the cyclosporine was that the mode used for analyzing samples was important. So general versus multi-purpose mode, especially because for cyclosporine, we found that these were bimodal peaks rather than single peaks. When you provide the method evaluation data, we would like to see the results be in the same parameters as for your pivotal study. So if your B conclusion is based on D50 and span, then it would be useful to have that as a metric for the validation. I think the next item is very fundamental to analytical method that you use, right? So any PK study on, let's say, tablet products, you want to use the validated method to analyze a sample. So that principle applies here as well. So make sure that your method development validation process captures the actual method that you're using for your pivotal study. I won't go into too much on the other ones here. Oh, what am I doing? Okay. For serial dilution, you will see the recommendation for this emulsion products. There is a recommendation for dilution study, and you will want to use the dilution factor where it's not fluctuating too much on the results. So that's important data to provide. And as with any analytical reports that you might submit for, let's say, tablet PK study, you please provide complete raw data so we can look at them and then evaluate for any concerns or whatnot. Oh, let me go back. And then for serial dilution study, we've seen situations where dilution factors evaluated were not different enough. So for example, applicant might have considered dilution factor of 10, 20, 30, whereas we would like to see magnitude difference in dilution factors. So then if they're too close to each other, it may not be meaningful information. For the pivotal study, let's see. Yeah, sometimes applicants report that there were 6 replicate measurements, but only 3 replicate measurements were used for a final B analysis. And in such case, that we might want to see what the explanation for not using all the replicate measurements. We've also seen that mean values were used rather than individual replicate values. So that's something to consider in your pivotal study. And then as a last part of the GSD study component, When you're analyzing the data, and especially the EMD and PB analysis, the transformation should be consistent with how you analyze the data originally. Okay, go to this one. Sorry. And then, so for the PSD study, the themes are very common. So please provide sufficient details related to the study so that we can understand how the parameters were selected. And then also, again, provide the raw data. And again, here, the information on the drug product batches that you're using for the test product as well as the reference product would be important. And for the pivotal study, again, provide the instrument setting. We've seen cases where applicant was repeating the sample analysis, but they were not consistent in how they handle those repeats. So in those cases, it'd be good to have SOP in effect that's specific, so then you're using objective criteria to determine whether to repeat the sample and how those repeats should be handled. If somehow you have to optimize your method, that might mean that you may need to redo the pivotal study, so that's something to consider. IVRT study. Xiuling has gone over extensively on a unique IVRT method that was developed in his lab. So I think that's a very unique situation. Of course, oftentimes applicants may use compendial USP method to conduct IVRT study. Again, details on the parameters selected, for example, depending on which IVRT method you're using, starting rate selection or the full rate selection would be important to know. And what variations have you tried to say this is the optimal method? If you're using a membrane, please include a membrane innerless study. If some applicants are using more than one membrane, then explain your rationale for why you're using more than one membrane and how that may not affect your study results. Again, using the consistent method throughout the study is important. One of the complicated, complexities and more red flag issue from our end is when the drug release is incomplete, or if there's high variability, then it's concerning. And those cases may mean that you may need to further optimize the method. If incomplete release is just a matter of adding on sampling time, that might be straightforward. But if it means your method is just never going to reach a complete release, then that's a more serious issue, I think. If you have high variability, then we might ask you to investigate on the reason for high variability. And then also, if we see more than 100% release, as with the dissolution data, sometimes we see that is something that we might wonder about. Why is there more than 100% release? Again, if you have these issues, that might mean you need to further optimize your method, and therefore you might need to repeat your pivotal study. Discriminating ability is, I think, a big challenge for the IVRT method, as discussed earlier. One of the issues that we run into very commonly is that we don't have sufficient details about these modified test batches. And sometimes when you have used a modified test batch, the method is not showing discriminating ability. So then we wonder, okay, so what does that mean? Let's see. And then as for how to analyze the release profiles, the commonly observed method is F2, but sometimes depending on the variability of the data, bootstrap F2 might be necessary. And of course, you have the freedom to propose another method to analyze the data, and we will consider that during the scientific review. For IVRT study, or related to IVRT study, saturation solubility will be something that you want to consider. But if you are evaluating the medium that's different from the pivotal study, then that's we consider incomplete information. Also, that's something that you want to consider. Yeah, and then pivotal study, again, please provide the details. I think we've seen other IVRT-related presentations today where the presenter shows schematic of the instrument setup, especially if you're not using the USP method. This is really helpful to have so that we can understand how this experiment is conducted. We have issued deficiencies asking clarification on whether you're using the 12 individual units or you're pulling the multiple units and using 12 replicate of the pooled sample, which we don't prefer. Of course, we asked for a variety study on 3 batches, each of the test and reference products. It's possible that maybe on the market there are not sufficient number of RLD batches available. In such case, then you can provide justification of and your efforts to collect additional batches, so that we'll consider that. Kind of repeating what I mentioned, reason for potential repeating IVRT study may be related to incomplete release or high variability, and also if the method is not sensitive or discriminating, that there may be another reason to have having to repeat the IVRT study as well. Again, for F2 analysis, the typical method we have seen for this profile comparison. Another part about IVRT studies, there's an analytical method component in addition to IVRT study component. Oftentimes in the study reports, we have this 2 information combined in one report, but it might be good to separate or tease out this information and provide the method validation data specific to the analytical method for our consideration. We have also seen cases where applicant used a 1-point or single-point calibration curve, but multi-point calibration curve would be preferred here. If somehow you're storing the sample after collection but before analysis, then provide the stability data to cover the storage condition and duration of those samples. And if there are any repeats, please let us know. And again, in that regard, it'd be good to have a specified and objective SOP for handling those cases. For global study or global size distribution related information, we have asked for mass balance study, and this is to measure and ensure that drugs that you're measuring in the retention media, release media, is actually the analyte and not the analyte that came through the membrane with the globule. So in that regard, you want to have an analytical method that addresses the key formulation ingredients that's measured in the mass balance study. 2 additional elements where we have asked for initial deficiency was related to retention sample. If you have any question in this regard, we do have a guidance in this regard, or if you need any other additional clarification, you can use a control approach to some of your question. And also, we have situations where OCs found some data integrity issue at the in vitro site, and so that was a basis for asking for a new study. So key takeaways. So I heard challenges related to Q1, Q2 sameness of the formulation. It is, I think, a starting point is if you have any doubt, then you can approach agencies through controls to confirm Q1, Q2 sameness of drug product. At least then you know that your product, your application is eligible for in vitro option. Be sure to provide detailed method development report so we can understand your story about how the product method came about. For IVRT method, as mentioned earlier today, discriminating ability is very critical for accepting the method. provide information related to the modified test batches that you used, and then make sure that your method is properly validated for use in the pivotal study. And having good documentation helps us to understand your method and your study, so then we don't have to issue so many deficiencies about details that were not submitted. And then finally, you can avoid reconducting the pivotal study and by making sure that your method was properly developed and validated. With that, here are some resources that you can consider. One of the earlier presenters talked about the PSG forecasting. I found the CRCG workshop a couple of months ago on the particle size measurement very useful. And also I mentioned some guidances that you can consider for reaching out to agency, either through the controls or formal meetings. For complex ophthalmic products, inherently, then would be eligible for meetings with the agency. So that's something you have for your consideration, unlike some of the simple ophthalmic solution products. And finally, I'd like to acknowledge all the bioequivalence assessment teams who worked on these reviews. Priti Jain and Shri helped pull these applications from all our database. Dr. Upamunshi provided feedback on my presentation. Dr. Partha Roy for his support. And finally, the workshop organizers for inviting me to give a talk. Thank you. And I'll take the questions at the panel session. Thank you.
Thank you, Huzeyfe. Next, we'll hear the presentation from Dr. Jessica Spires, Simulations Plus. Welcome.
Hi. Hello, I'm Dr. Jessica Spires. I'm a principal scientist at Simulations Plus. Thank you so much for letting me present today. So I guess we can kind of get right into it. Really, I would say one of the major gaps that we often have when we're looking at these ophthalmic formulations and trying to figure out what's going on is we want to know what these local concentrations really are when we apply these to the human eye. You know, typically we're trying to treat some particular ocular condition. You know, ideally we don't have anything reaching systemic circulation. Often we don't have anything measurable reaching it. You know, so if we're really trying to reach, you know, these ocular tissues, you know, we may not have any way to measure those in vivo in the human eye. Or even if we have the ability to do it, it may be very difficult. You know, other than the tear film, you know, people don't want needles stuck in their eyes. And Dr. Lerman mentioned, you know, in some cases people will have complex conditions, so it's difficult to recruit for studies and things like that, okay? And this can also make it difficult to understand what's happening with the efficacy as well. You know, are we achieving the target concentrations of local tissue of interest? Do we have some mistake about, you know, how much we need to get the effect that we want, things like that, okay? Of course, when we have the preclinical species, oftentimes we can take more extensive measurements, we can be more invasive and so on, you know, but then when we're switching species and we're trying to figure out what's happening in the human eye, you know, is the question, you know, how close are we? You know, how similar is it really going to be? So in this case, I'll be talking about brinzolamide as well. I think there's already been some discussion of the effect, so I won't go into too much detail. So brinzolamide is going to be acting to reduce the aqueous humor generation, okay? And of course, the reason why we're interested in this is to treat conditions such as glaucoma and ocular hypertension, because having sustained high IOP can potentially lead to blindness and things like that. So we have the marketed product, Azopt, which is, as we mentioned before, is a 1% ophthalmic solution, typically going to be administered to patients 3 times per day, okay? So in addition to having the information about the compound, unfortunately in this case, we also had information about the particle size distribution and things like that for the Azopt. Generally speaking, for these ophthalmic formulations and for these complex formulations, we do want to have a good understanding of those key quality attributes. And in this case, for azopt, again, knowing this particle size distribution was really key in really understanding the formulation and how it was affecting. Oops. So what kind of experimental data did we have available in this case? Okay. In this case, we had local concentrations in several different parts of the rabbit eye, including the cornea, conjunctiva, aqueous humor, which of course is the one we're most interested in, as well as the iris ciliary body. But in the human eye, in this case, we actually only, and then we also had some of the PD effects as well. In particular, in this case, we want to know how the IOP is going to be changing over time, as we apply the drug. But in the human eye, we don't have any kind of ocular PK in this case. In this case, we only had the measurements of the IOP reduction. Okay, so the question is, how can we bridge this gap? We know a lot about what's going on in the rabbit eye. We know a little bit about what's going on in the human eye. How can we make all of this line up? Okay. And if you know anything about Simulations Plus, you're probably not surprised. For us, the answer is going to be looking at PBPK modeling. Okay. So in this case, we have a model that's describing the different tissues of the eye. As you can see, it's very complex, probably one of our most complex models. I won't go into too much detail about it, but some of the things that we incorporate here are going to be some of the physiological aspects, what are the volumes of the different tissues of the eye, how do they contact each other. We include things like the diffusive flows, the various convective flows of the eye, and we also take into account which parts of the eye are going to be vascularized and which ones are not. Okay. And then this can also be connected to PBPK models of the rest of the body to describe the systemic distribution and clearance. So the first thing that we want to do in a case like this is to create a baseline model of the PK in the rabbit eye using the data that we have. Okay. So typically we're going to have some initial estimates of key compound parameters like the permeability in the different parts of the eye. But typically, we do need to have some kind of data to optimize those values. Our predictions are based on a very small sample set. Typically, we expect that we're going to need to make some differences there. So we can use data like this. We have data in several of the different tissues to optimize these types of values. And again, in this case, you can see the check mark next to aqueous humor because that's one that we were particularly interested in. So once we kind of set up that baseline model, I think in that case we're using some of the data that was generated by the FDA. Next, we wanted to validate that model. And in this case, we were combining this data from the FDA that we used to create the baseline model, along with a lot of published data of the azopt applied to the rabbit eye in a lot of different studies. Something you'll notice here, first, if you look at the points here that are representing the experimental data, you'll notice that there's going to be a lot of variability. And I would say that that's going to be very common when you're looking at these ophthalmic cases. The other thing that you'll notice here is that the lines, you'll see that there are a lot of different lines here, and those are reflecting kind of virtual population, where we have kind of stochastic, physiologically reasonable variation of the different physiology of the eye. Okay. So I would say this is something that's really important to keep in mind when you're looking at these ophthalmic models. It's very common to have high variability, and instead of really trying to say, okay, I need to hit exactly this point, exactly that point, we need to start to think about, what's the physiological range that we see with the experimental data that we have, and then when we start to model the physiological range, are we correctly matching that range? Okay. And you can see that in this case, across all of these different parts of the eye, we're generally getting a pretty good compromised model. Okay? And we're getting a pretty good representation of the physiological variability that we see. The next step to this is that we want to link our PBPK model to a PD model. And particularly, we want to be modeling the IOP reduction in this case. I think on our side, since the IOP is very complex, we actually ended up making kind of a special custom IOP model. It's also accounting for the fact that you have circadian rhythm of the IOP and things like that. Okay. In particular, when we did our initial modeling of this, we were particularly looking at modeling this in the Dutch-belted rabbits. Okay. We're also able to link that specifically to that local PK concentration in the aqueous humor. So we're able to link the local concentration to the PD, so we can really have a direct effect there of what we expect is happening directly based on the compound. Okay. So we started off, of course, the one that you see on the left here is going to be the 1% dose, so that's going to be representing azopt. You can see that we had a very nice match to this. And then we also wanted to apply this to some of the other doses as well. So you can see the 2% here is also fitting very nicely. For the 0.5%, we do see a bit of an undershoot. I think, in this case, we're really focused on azopt, so we weren't too concerned about this. But I would say if this was a case, where we really needed to know what was happening, as we're changing the concentration, this is another one of those cases where I really want to start looking at the virtual population to see, is it just that that we're not matching these one little points, or are we kind of systematically off? But in this case, we said, we're happy with it. Let's also apply this to the other cases for validation. So we had a number of studies also looking at this IOP reduction in the New Zealand rabbits. And one thing that you'll notice here is that we did actually have to make a small change in the PD model. And the reason for this is because there were actually some baseline IOP differences, before any drug is given, between the Dutch-belted and the New Zealand rabbits. Okay, so that's something that you sometimes have to think about as well. It's not only the PK, but there can also be physiological differences that are potentially affecting the PD models as well. Okay, so once we took that into account, there was this baseline difference between the two breeds of rabbit. As you can see, we're able to get a really nice match. So on the left here, you can see comparison with the aqueous humor, and then on the right, you can see how, that we're really getting a good match to the variation that we're seeing in these New Zealand rabbits. Okay, so at this point, we're pretty happy with the model. Okay. So of course, ultimately though, we really want to know, again, what's happening in the human eye, okay? So how do we actually do that extrapolation, okay? So first step, obviously, is you go to the little drop-down menu in the program and change the model to the human eye, okay? And this is going to be taking account of all of the different physiological differences that we expect between the rabbit and the human eye. So again, these things like the volumes, the surface areas, the differences in vascularization and things like that, okay? For these parameters like the permeabilities, typically we're going to continue to use the ones that were optimized by the rabbit eye model. And in general, we've had a lot of success with this approach over several different compounds. These permeabilities and so on seem to carry over very well between the rabbit and the human eye. Okay, so we keep those same values. Okay. And the other thing that we needed to do in this case as well is because of the intraocular pressure, and we were also looking at very long-term studies in this case, I think going over weeks or months, we also needed to establish the baseline circadian rhythm of IOP in the humans before it's affected by brinzolamide. Okay. If we've been doing longer-term studies in the rabbits, we also would have wanted to consider that. So again, the first step was to set up that baseline model of what's happening with the intraocular pressure. Fortunately, you can see that we had a number of different studies here. Again, note there's high variability in the experimental studies that we're able to match with the virtual population model. Okay. And then finally, we can really start to look at modeling the IOP reduction in the human eye over the long term. So it's a very busy slide, I know. So on the left-hand side, this was looking at cases where the AZOP was given BID twice a day. On the right side is showing the studies where it was being given 3 times a day. Okay. And then at the top part, you can see kind of the full scale. And then at the bottom, you can see kind of some close-ups of the different days where they sampled, day 8, day 15, and so on. And you can see that even going out to 90 days or 180 days, we're getting a good match to the experimental data, and we're also getting a good match to the variability that we see in the experimental data. Okay, so this is a point where we'd say, we're very happy with this model. We were able to build it and validate it in the rabbit eye, and apply it to the human eye, and get a good result. So just to summarize, with a close attention paid to these physiological differences between the rabbit and the human eye, it is possible to extrapolate these ocular PBPK-PD models from rabbit to human. Okay. And two things I would particularly point out here for brinzolamide, and also really for any compound that you look at. First of all, having a careful consideration of the physiological differences, not only as they relate to the PK, but also as they potentially relate to the PD. There can also be differences in these between the species. Okay. And secondly, it's always very, very important in these cases to consider the variability. It's very typical to have high variability in these cases between subjects, between studies. Don't be trying to chase a single point, when in reality, it may be that once we take that variability into account, that we have a very good model. Okay. So of course, the next steps would be, what else can we do with a model like this? I think Dr. Lerman really spoke about this very well in considering the generics and the regulatory space. We can use this type of model to support waivers, to support that these two formulations may be bioequivalent, as a support. In addition, in the next two in the next two presentations as well, we'll be talking about how we can use the in silico modeling in combination with not only the in vivo data, but also the in vitro data, so that we can use this to inform our models, to support our models, to extend our models. So you can look forward to that. Okay. So thank you all very much. At Simulations Plus side, particularly want to thank Lexi Lumerdi, who did a lot of the modeling work for this project. This was fully funded by the FDA, so thank you all so much for that. And at the FDA, I also particularly want to thank Mingliang 10, who really helped us a lot, as well as everyone else who was able to contribute. So thank you very much.
Thank you, Jessica. Next, let's welcome Dr. Mingliang Tan from the FDA.
Works like this. Thank you, Anne, for the introduction. Good afternoon, everyone. My name is Mingliang Tan, and I'm a senior pharmacokinetist in the Division of Quantitative Method and Modeling, Office of Research and Standard, and OGD. It's my great pleasure to present in this session. My talk today will be focusing on ocular PBPK-PD modeling to explore in vitro bioequivalence approach for brinzolamide ophthalmic suspensions. Yeah, thanks, Jessica, for the nice presentation, which make my job much easier, so I don't have to go over all the background information here. First, I would like to state that this presentation reflects the view of myself and should not be construed to represent FDA's view or policies. My talk will start with some background information and the challenges in developing generic ophthalmic suspension for brinzolamide, followed by PBPK modeling effort to explore in vitro approach for brinzolamide ophthalmic suspensions, which include PBPK-PD model development and validation in rabbit, model interspecies extrapolation to human, and then model validation through literature data, followed by modeling efforts to understand the impact of formulation in vitro characterizations on the in vivo performance through FDA internal data submitted by ANDA applicants. And finally, VBE assessment for approved ANDAs and the sensitivity analysis. As you may be aware of, an in vitro B option has been recommended for multiple ophthalmic suspensions, for example, API as steroid or non-steroidal anti-inflammatory drugs or NSAIDs. However, an in vitro-only approach has not yet been recommended for ophthalmic suspensions product that is indicated for intraocular pressure or IOP reduction. Because IOP reduction products such as brinzolamide ophthalmic suspensions are associated with high risk due to long-term or chronic use, as well as high risk to the patient caused from uncontrolled IOP by a non-therapeutically equivalent product. So the current or the historical thinking is that an in vitro approach may be recommended for IOP suspensions product when a better understanding of how their in vitro characteristics affect in vivo performance. For example, when studies and data demonstrate a good in vitro-in vivo relationship. Here is the current product-specific guidances, or PSG, for brinzolamide ophthalmic suspensions. As you may see, current PSG recommends comparative clinical endpoint BE study to demonstrate BE between test or T and the reference or R products in patients with chronic open-angle glaucoma or hypertension, ocular hypertension. So comparative clinical endpoint BE study can be used to assess local concentrations indirectly, but this study has their own challenges, such as that study may be confounded by the degree of patient disease severity, variability in measuring drug response, and the lack of meaningful discriminations to formulation characteristics and changes. Then the question is whether an in vitro B approach can be an option for brinzolamide suspensions. In order to proceed with an in vitro B approach, we know ophthalmic suspension needs to be well characterized, firstly in vitro setting, and then, mostly importantly, the impact of suspension formulation in vitro characteristics on in vivo performance needs to be understood. In this regard, physiologically based pharmacokinetic or PBPK modeling approaches may be used to understand and demonstrate the in vitro-in vivo relationship. Before we go into the detail for the PBPK modeling, let's first take a look at the related data available for modeling brinzolamide suspensions. For the brinzolamide suspension critical quality attribute or CQA data, multiple publication and FDA-funded research available, as shown in a couple of previous slide, presentation already actually. In the preclinical space, there are some rapid PK and intraocular pressure or IOP data available from both literature and FDA-funded research. For human data, here there is no ocular PK data available for brinzolamide, but there are some human IOP measurement available in the literature, as Jessica just demonstrated earlier. Lastly, there are some FDA internal data available, including human IOP data and the formulation CQA data submitted by the applicants. Based on the data available, we proceeded with the PBPK-PD modeling for brinzolamide ophthalmic suspension. Now, let's first take a look at the PBPK and PD modeling effort, which was again trying to create formulation sequence with brinzolamide, ocular PK and PD mechanistically use data from 3 ANDA submissions. The plan was to start with extrapolating the validated rapid PK/PD models to human, and then validated the model using literature data. Yeah, thanks again to Jessica for explaining this part in detail a moment ago in her previous presentation. That again makes my job much, much easier. Then followed by incorporating the formulation sequence data, mainly particle size distribution data, and the viscosity data for suspension submitted by 3 ANDA applications into the validated model to predict human ocular PK and the IOP. The predicted IOP was compared to submitted IOP measurement in 3 ANDAs for model validation purpose. Then predicted the human ocular PK data were used to conduct a virtual BE or VBE study to see if the VBE meets the BE criteria in terms of PK in ocular tissue for the 3 approved ANDAs that were again demonstrated to be BE based on IOP or PD measurement from comparative clinical endpoint study. So the key question here is whether the human ocular PK meets the BE criteria for 3 ANDAs that were demonstrated to be BE based on IOP or PD measurement from comparative clinical endpoint BE study. Now let's take a quick look at the baseline PBPK-PD model again. Jessica just did a great job in presenting this part and already, but I still would like to give a quick recap on the base model development and validation in the next couple of slides. The brinzolamide baseline model was developed and validated first in rabbit, as there were some ocular data available, relatively rich data from literature and FDA-funded study, including brinzolamide concentration measurement in rabbit tissues such as cornea, conjunctiva, aqueous humor, and iris cellular body, as shown in the plot. Overall, the simulated PK profile will capture the observed data as shown here. Then the validated rapid PK data was extrapolated to PBPK-PD model to predict the rabbit IOP in rabbit eyes. The predicted IOP was compared to the experimental measurement from various studies, from both literature and FDA-funded study, shown here. Then the validated rabbit PBPK-PD model was extrapolated to human by accounting for the interspecies difference in anatomy and physiology. Then the extrapolated human model was validated against literature IOP data. Shown here was a simulated human IOP compared with the corresponding measurement after twice-daily dose of brinzolamide suspension for 180 days. Shown here was a simulated human IOP compared with the corresponding measurement after 3 times daily dose of brinzolamide for 90 days. Again, here is a model plan for the 3 approved ANDA. Up to now, the baseline human PBPK-PD model was developed, and that will be used to simulate PK and PD for 3 ANDAs. By utilizing in vitro formulation sequence data, including particle size distribution and viscosity from ANDA submissions, the predicted IOP will be compared to the IOP data from the comparative clinical endpoint B study through ANDA applications. Then the predicted human ocular PKs, which is not measured again, but simulated, were used to conduct a virtual BE to see if the VBE meets the BE criteria in terms of the PK in ocular tissue for the 3 approved ANDAs. Finally, sensitivity analysis on the drug ocular tissue exposure to formulation CQS will be studied. Shown here is a time course of simulated human IOP compared with corresponding measurement from one ANDA submission after 3 times daily dose of brinzolamide suspension for 42 days. Bottom left is a snapshot for day 14, and bottom right is a snapshot for day 42. Shown here in the table is the virtual BE assessment result of Cmax and AUC in aqueous human tissue for 3 approved ANDAs, ANDA A, ANDA B, and ANDA C. As you may see, the TR ratio and the 90% confidence interval of Cmax and AUC between RLD and the 3 ANDAs are all within the 80 to 125% BE range, suggesting passing BE outcomes in local aqueous human tissue from local tissue PK point of view. Again, those 3 ANDAs were approved based on the IOP data from comparative clinical endpoint studies. Shown here is the sensitivity analysis of particle size distribution on the drug exposure in aqueous human tissue. The left panel is the result of Cmax, and the right panel is the result for the AUC. The result indicates that the drug exposure in aqueous humor decreases as the particle size increases as expected. Shown here is the sensitivity analysis of viscosity on the drug exposure in aqueous humor tissue. The sensitivity analysis shows the exposure in aqueous humor reaches a plateau when viscosity reaches some value. For example, here is after 50 centipoles. So in summary of this research project, first, PBPK-PD model prediction are in agreement with the human IOP data from 3 ANDA submissions through clinical endpoint studies. VB assessment demonstrated BE in aqueous human tissue based on the predicted drug tissue concentrations that were used to IOP prediction for the 3 approved ANDAs. So lastly, sensitivity analysis suggests that aqueous human tissue is sensitive. Exposure is sensitive to the particle size distribution, while may be less sensitive to the viscosity for brinzolamide after viscosity reaches a certain value. Here is a list of references for the experimental data that were used in my slide for your reference. Finally, I would like to acknowledge my colleagues at ORS, OGD, FDA for their support for this project: Angie, Eleftheria, Andrew, Lucy, Yan, Lei, and Rob. I also would like to acknowledge Maxim Nemader for his great work in developing this baseline model, Jessica Spire and Veria Lukikova for the collaboration from Simulations Plus. Thank you all for your attention.
Thank you, Mingliang, for the presentation. And last but not least, let's welcome Dr. Carrie German from CFD cooperation. Welcome.
Okay, I get the privilege of taking us home today. So I want to first thank the organizers. I appreciate the opportunity to be here. I'm very excited. I'm Carrie German. I'm from CFD Research Corporation, as they just mentioned, and I'm very excited to share our work in silico modeling of ophthalmic suspensions going from in vitro to in vivo. So I know we've gone over the challenges very well previous to this. The previous speakers have done an excellent job, so I won't belabor that point. I think when we saw the questions of challenges, we thought, how can we help model, how can we help answer this question and answer the challenge? And where can we help? So we kind of saw the problem in these 4 buckets that you guys see here, and we wondered if we could help in multiple different buckets. So on the compound characterization side, a lot of that comes from the formulation, people who are developing the formulations. We see our ability to help primarily on the right sides in the in vitro release testing, which has already been discussed earlier today. ADME studies, a lot of that, it could be penetration, looking at ex vivo tissues, looking at functionalized organ on chip devices, things like that, and those can vary based on the operation parameters. And then finally going into in vivo PK/PD tox, and that can be in animal studies or in clinical trials. So where can we fit in, and where can in silico modeling help in this process? So after we decided we wanted to work in these different buckets, the next question was, what type of modeling approach did we want to use? So you can see kind of in the bottom, you always have this paradigm of complexity versus cost. We can start with a model that's really driven by data, that's like a 2-compartment model. Non-physiological, we can move it up into physiologically based pharmacokinetic models like the ones that were presented previous to this presentation. We can go into very complex high-resolution models that show distribution and complete anatomy of the eye or of these in vitro systems, but you're trading off, what do you gain? So these are going to take longer to run. When is it appropriate to apply these high-resolution models, and how can they help? So we asked ourselves those questions as we went through each of these different pieces. I thought the easiest way for us to demonstrate this was to look at a specific compound. So all of these will be applied to dexamethasone, but have been obviously applied to different compounds as well. So the physicochemical properties have been discussed previously, so I won't go into those. Those have been collected from literature, but we are looking at different formulations. And so some of those that you will see here on the left, those were formulations that were created by the University of Eastern Finland team and provided to us with different particle sizes and viscosity. And then you can also see the characterization they did for Maxidex as well. So we will use all of these properties as inputs to our simulations. So moving from the characterization into the in vitro testing systems, release testing systems, we started with a slidalyzer. You can see the schematic of what that looks like and what the volumes look like for that. There on the top left, it is a small system and it is stirred, so we felt that those operation parameters were appropriate for an ODE-based model or a well-stirred reactor type approach. You can see the model down there in the bottom left. This transport includes species transport, or this model includes species transport as well as dissolution as needed. You'll see here that we start with solution, so our approach is very much to start with the simplest case and build the complexity. So if we can understand transport through the membrane, then we want to add dissolution. We don't want to do both at the same time. See if we can understand one before we jump into the other. This model also includes a membrane fouling that was discussed earlier, and then pH contribution. So as Jessica really pointed out, you start with your first try, and then you see what physics are missing. So we weren't matching well with the first go through, and so we looked at microenvironment pH contributions as well as membrane fouling, and we were able to eventually match very well with the experimental data based on the input from the experimental setup as well as the operational parameters. So the next step that we wanted to look at after we could look at the solution going through, now we want the complex formulation. So next we looked at those 5 different dexamethasone suspension formulations in the same setup, but now with the different properties. So we put those properties that you saw on the previous slide into our simulations. And you can see here the simulation tool for those different simulations and how you can select through the different options to run that model through our user interface. But through running those and using the transport properties that we had from the previous solution simulation, as well as these properties for the formulations, you can see our different errors. Our mean absolute error was less than 10 for 4 out of the 5 formulations, which we were very excited about. So being able to translate from solution to suspension was successful. But you can put this into a bunch of different apparatus, right, as we heard earlier. So this was another study we did with dexamethasone and looking at it in the USP4. This was a different formulation. This was taken completely from literature. So we set this up in what's called our quasi 3D, so it solves PDEs in the direction of flow and approximates in the axial direction. So we can solve this whole flow loop for the USP4 setup. So this includes spatial resolution. It includes diffusive and convective transport contributions through the system. We set this up just like we would for anything else. We'd start with a solution. We use the operation parameters. Something interesting about this particular simulation was that the experimental data showed no impact of operational flow rate through this system. And so we wanted to make sure we could validate that when we put a solution into the system. And you can see we were able to match very well, and you really can't see all of those simulation lines, because they are overlapping and we don't see an effect when we change the flow rate from 8 to 16 mils per minute. Similarly, moving into the suspension, we're not changing the parameters. We're using formulation parameters from literature for Maxidex at this point, and we use the same data that we learned from the solution in the suspension. So we're adding also in particle dissolution physics. We're using a modification of the Noyes-Whitney equation for that. We can include particle size distribution, so looking at a poly versus a monodispersed population, and in this case, we're looking at a monodispersed, but we could see how that effect changes the simulation profile, and then also changing those formulation parameters. So you can see how the model was set up above the plot, and that we do slightly overpredict, but we are definitely capturing the trend, and we're right there at the edge of the standard deviation of the data. So just another way that we looked at this, we wanted to understand a little bit better. Again, spatial resolution, is it that important? So we looked at this same setup with a 3D wedge and connected to our Q3D model rather than just a Q3D model of the USP4 apparatus itself with the adapter. And you can see in these that we can definitely detect a spatial resolution impact specifically in that adapter region over the 3-hour simulation time. And then we were able to replicate those results from the Q3D case using this 3D model. So if it depends where we're taking these samples, but it could be very important for us to see that this mixing doesn't occur until later on in the experiment. So that may be important when you're collecting some of this data from the reservoir rather than the flow cell itself. So moving from in vitro and then to ex vivo, we want to understand permeability. A lot of these are difficult to obtain values, but we have tried to, again, simplify the problem. Again, working with the University of Eastern Finland on this project, we were trying to look at the effects of excipient addition. Specifically in excipients, when we say excipients in this case, it is the viscosity modifying excipients, and then before, obviously, we need to have some excipients in there to dissolve the dexamethasone. So those are in there when it says without excipients, but with excipients is the viscosity modifier. So this case, we're using the Xiuling chamber. We have an ODE and spatially resolved models, but they gave very similar results based on how we set those up. So we have included species transport, partitioning because of the tissue inclusion, and volumetric sampling into this setup. You can see the parameters that we've used from previous simulations can be used again for these. So some of those transport properties can be reused here. We input the experimental parameters for dose. We needed to know the size and the volumes that were used, and then the media composition as well. But you can see here for the predicted versus the experimental, we add in the variation for the plus minus on some of those particles, for some of the apparent permeabilities. In this case, we are using solution since that's the only part of this that goes through to the other side. And that matches really well with the experimental data. The next step for us is we have a method of calculating apparent permeability through the corneal tissue. And so we then compare that with the data that we got here, or that was experimentally determined, and then here. So we do need more data and more ways to modify that to account for excipients that are not just viscosity modifiers, because we can account for viscosity in our transport, but there are other ones that contribute to solubility changes that there's more data that needs to be fed in. But that's a next steps for those models. Finally, in vivo modeling, we're looking at PK, and these can definitely be linked to PD. We previously were using these 2D models of the eye that were limited because we could only do in vitro, or we could only do like intravitreal or intracameral injection type modeling approaches. We were missing that tear film. The flow profiles don't play together nicely in 2D, so that's why we needed to find another solution. Multi-scale modeling was our approach to do that. So we were able to actually connect 2D models to resolve flow and temperature and pressure profiles in the eye, and link that with a dynamic tear model, which you guys can see is a compartmental model for both the human and the rabbit. These have been, you can think of them like a 2D slice of a histology slice. They are a planar model, not a 2D axis symmetric model, and we look at the 2D performance alone versus 2D plus ODE on the next slide. These do include species-specific anatomy to help support translation. And then you can see the validated pieces there below. We validated the rabbit model is within normal IOP. For this condition, we are doing normal. We can do diseased as well by changing boundary conditions. You can see that for the rabbit, it's within normal IOP, and then the flow rates have been validated based on velocimetry profiles as well. So getting to the data, we can speed this up. I told you there is a trade-off, right? These can take not minutes, not seconds, not minutes, but if you run them with full flow and species transport solutions, it can take hours. So we wanted to figure out how to speed that up. If you assume a steady state condition that the flow profiles aren't going to change, you can use those velocity profiles over and over at each time point and don't have to solve. So it makes them much faster. So we use that to decrease our computational costs. We are still including species transport. We have convection and diffusion. We are including partitioning as well as elimination from those tissues that have blood flow, and it's based on those blood flow values that are included in there. Again, we're using those from the ex vivo studies. We can use those permeation constants. We've done them for cornea and conjunctiva and are now looking to do iris ciliary body going forward. And we can use those to plug in as values for this modeling effort. So you can see the setup. What I'd like to point out is the simulation of the 2D only, and the simulation which just says simulation is 2D plus ODE. And you can see that without adding that tear film on, we don't have the correct elimination component because we don't have that concentration-driven elimination from the cornea, pulling from the cornea into the tear film. So this was a big step for us to be able to validate this model on in vivo data. This was specifically for an intracameral injection. So we didn't have to have the tear film, but it was definitely worth being able to show that. In the last application, we did in vivo modeling and application to the tear film now. So that's the whole reason we wanted to do this, was to be able to do a topical application. And we use the exact same setup, the exact same permeation values, all of that from the intracameral injection studies. but this time we're adding it, Maxidex, to the tear film part of the model with volume dynamics for the volume installation. And so we add those to the tear film compartment, and you can see that we're able to match very well with the tear film data from, again, from the University of Eastern Finland. And one thing I just wanted to point out is yes, you can see the movies and see the transport visually of the species through the model, but you can see how different that gradient is throughout the aqueous humor. It's not, if we're really worried about taking values for the values at the trabecular meshwork, these models might be really important for getting very localized concentrations. So other application areas, we've looked at lots of different in vitro setups. We've looked at the effects of particle size. So these actually, I think, we were looking at setting up some of Dr. Xiu's experiments. So those looked very familiar when he was going through those. But we can look at how those affect distribution through the system. We can look at operational effects. The RPM obviously will be very important, and we can see how that affects mixing, velocity profiles, and vectors inside of these types of simulations. And then the other part kind of echoes back to what I just said. We can see with spatial resolution, say across the aqueous humor or across the lens or across the vitreous humor, we can set up these lines to see how the concentration profiles vary with respect to space. I already mentioned that these can also be used for interspecies translation, and that we do see very different profiles if we apply the exact same concentrations to the tear film for human versus rabbit modeling. And with that, I would like to this was all funded by FDA as well, so thank you for that, and thank you for this opportunity. Thank you for the collaboration as well. UEF for the experimental data that was extremely valuable in being able to validate the models. And then of course the CFD team. Oh, and CFD is computational fluid dynamics, if you did not know it. If that's on your bingo card, there you go. Thank you guys.
Thank you, Carrie. And with that, a huge thanks to all the speakers, but now job is not done yet. I would like to invite all of you to the podium, and we're going to start our panel discussion Q&A session right away. Okay. So with that, I think we can officially start our panel discussion. And Andre, please go ahead.
Thank you, everybody. I really appreciate all the talks of the afternoon. I have a question that is more focused towards the talk that Dr. Lu gave, but it's actually applicable to the talk that several of you have given, which is related to this relationship between in vitro, ex vivo, and in vivo data. That is something that we have been striving a lot to do, but more often than not, we find ourselves seeing data in which it appears that formulation differences that we see in vitro may not necessarily translate to the data that we see ex vivo, and much less to the data that we see in vivo. I was just wondering if Dr. Lu, as well as other panel members, could comment on that and how you see the implications of those findings from a regulatory perspective. Of course, keeping that in mind that these are preclinical data, but they do inform our thinking in one way or another. Thank you.
Thank you, Andre, for the great questions. We keep thinking about this ourselves as well. So in vitro, we do that test, and I think it's more towards discriminating formulations to understand the differences between formulations. But our sense is it's oversensitive. It is very sensitive. And yes, we want to have a sensitive method first, and then getting this, if you are able to use this sensitive method and still see the similarity, and then I think we can guarantee in vivo that the performance will be the same. And then it gets to ex vivo, we see that as a good transition bringing to biological environment, but still it does not have the complexity and the variations is much less. I think it was very, very consistent into sample to sample consistencies. So we see that as a good transition to evaluate, at least from our comparison of tobramycin and dexamethasone, we see that for hydrophobic drugs, it gets better correlation and better prediction compared to hydrophilic drugs as a tobramycin. So, and then going to in vivo is a bit complex, and the variations is for struggle that we had. And again, based on the drainage and retention, especially for ointments and retention, we see that's an important parameter to consider, and that retention is related to viscosity of the ophthalmic ointment as well. So I feel like the sense is seeing all the differences between what go back to the goal, what are we looking for? So if we are go back to even Xiaoming's point, if we want to understand the differences between formulations, we try to change a bit, and then we are able to really get very sensitive evaluation of these formulations, and then we can guarantee in vivo performance will be better. So give us a bit confidence into this formulation characterizations.
Thank you.
I might just add on real quick. I think the computational modeling could also help, with some of those where you don't know why hydrophobic versus hydrophilic are those differences. I think computational modeling could definitely lend some insight there.
Yeah, we would love to do more of that kind of collaboration. Yeah.
So I have kind of a similar question. Dr. Liu showed how some of the in vitro methodologies can be quite discriminating, and in vivo, you might have bioequivalence or apparent bioequivalence, even though that in vitro method was discriminating. So that got me thinking, if I was a generic company, maybe I would just do the BE approach, because it might be easier to show bioequivalence with the in vivo test than the in vitro test. But then, in Dr. Chung's presentation, he mentioned all the difficulties sometimes of doing the in vivo bioequivalence study. So my question is, how does this shake out? Is it more common to do the in vivo bioequivalence? Does that end up being more feasible? Or are the in vitro approaches in general more feasible? From your experience over years of doing many of these.
So I can start. So in vitro, yes, you have these sensitive methods and you feel like you're able to see the differences, but you also are able to get the similarities of certain formulations. And not like we are now able to get two similar bioequivalent in terms of in vitro method that we are able to. So I would say in vitro methods is still much easier compared to in vivo evaluations, right? But also, again, we have more confidence having all these sensitive methods in vitro.
Since I've not seen application that contain both in vitro and in vivo studies, I'm not sure that I can address. That said, for other products where we saw that in vitro study failed and the in vivo study passed or vice versa, and if we saw that in an application, we would definitely have a question, right, whether this is truly bioequivalent or not. And so we might seek additional clarification or have questions to the applicant to better understand and whether there's clinical relevance on in vitro data that might have failed versus, let's say, clinical endpoint study that did not pass, and then maybe the clinical endpoint was not appropriate. So there are various possibilities, right? So I think those are the possibilities, but again, I have not run across that specific situation, so yeah, thank you.
So in vitro model you showed in the Tobradex, that in vitro model, as per the bioanalytical guidance, requirement of the validation meet the requirement, because there is a little bit challenging to validate the method to go in the real performance of the bioequivalence in the certain dosage form like the Tobradex. So what is the approach you put in the in vitro comparison for the validation purpose?
So let me see if I understand the question correctly. So I guess, there's always trade-off, right? Sometimes it might seem like maybe in vitro approach is easier, but if you run into complexities, let's say, related to demonstrating discriminating ability, then there might be a bottleneck. On the other hand, if let's say you're to pursue a consumer study, you know that this will be a very costly study and data analysis is complicated. So you have to consider from your own company's priority, what is the, if do you want to get the application through us for generic and to spend the money, or do you want to find maybe more feasible approach and less costly approach to get approval of the product? So I think there are many factors to consider, but I understand that sometimes validation or even developing a method can be challenging. And I think Xiaoming's case actually presented a good example where traditional IVRT method was very difficult, and this ophthalmic products, you're limiting the volume of the drug product to use as well, right? So I think their method tried to find out ways to demonstrate bioequivalence using the IVRT method and see, what's feasible in that angle. And so I think his group provides good example of a situation or consideration.
Yeah, just one thing, today's talks focused on what's in the bottle. Tomorrow we're also going to talk about the bottle itself. And so sometimes those things, the container closure system can affect the delivery of the drug too. And so the discrepancies between in vitro and clinical outcomes could be explained potentially with the amount that's applied, et cetera, for these products. Thanks.
Back to your question, I think another example probably would resonate with your comment is what Romit touched upon briefly. For instance, for tobramycin ointment, there are two different APIs in the product, and currently we are fully aware. We recommend you characterize both APIs particle size, and for that particular study, how you validate and how you compare. So here's honest feedback. Honest feedback is, we try our best to do certain research, right, to enable our development of alternative approach, but we don't have the resource to really get down to every single recommendation in terms of the true feasibility or how challenging it could be in reality in terms of the execution. So for that particular one, we actually had debate internally, discussion in terms of what is the most reasonable scientific approach at the time. And we decided, okay, still roll out as just assume it's feasible, it can be done, which we know is feasible. We have done some study, preliminary study to know there is feasibility, but then feasibility doesn't translate to easy execution. So for those circumstances, our PSG is really a work in progress in almost most cases, right? It's not final. So another way is we are seeking feedback from companies. So we often, this kind of scenario, we are often also testing the water to see how company reacts to our recommendations. And if you are really struggling with certain recommendations with strong rationale or evidence to show us what may be the more reasonable alternative, we are very open to that. And Romit, you probably could elaborate more if you would like to on this particular.
Method feasibility is a one aspect of the develop the method, right? But when you want to prove using the in vitro BE approach, you need full validation as per the guidance. And certain model in the Tobradex particular, there is do different kind of the drug substance in ointment, my matrix, right? To validate that method in a discriminating power is very, very challenging for the industry. So in that case, right, to prove the discriminating power as well as the mass balance and selectivity to as per the guidance is a little challenging.
Yeah, we appreciate that feedback, and that's something we can further discuss in a small group discussion. And I would like to actually welcome our online panelist and speaker, Dr. Vatsala, and thank you again for making efforts to pre-record your presentation, and welcome here.
Thank you so much.
If we have online question, we can take this opportunity to address.
Okay, actually there is one specific question. It said, okay, for ophthalmic ointment, and some applicants, they are doing like F2 statistical approach, but there is a USP 1724, which also like have a recommendation. It's use TR slope. So the company just want to know whether like they should just go with F2 or like follow USP. Like,
actually, we use SUPEC guideline. We use the SUPEC for that SUPEC guideline for comparison by equivalence. We don't use F2.
SUPEC SS.
Yeah. Yeah, for ophthalmic ointment.
It's old though. It's like 30 years old.
Yeah, I think that's a really good question. And that's something, again, we are aware. So the other alternative you mentioned, I mean, the audience mentioned in the question is a well-established practice for topical semi-solids. From formulation design standpoint, we are aware and agree there is no fundamental differences between a topical ointment versus an ophthalmic ointment. Although from the context of use, those things, there are differences. So right now, our general approach or expectation is really up to the applicant to decide and justify the statistics to demonstrate the similarity in the in vitro drug release profiles for ophthalmic ointment. The challenge for F2 is often the release is incomplete for ointment, but then so for certain circumstances, we have also accepted F2 analysis based on incomplete release with adequate justification. But this is something internally FDA are having ongoing expert discussion, and hopefully we'll have more clarity in our future PSGs. We'll try to give more direct guidance on those aspects.
And I might clarify that in my presentation, the ANDAs I looked at were gels, emulsions, and suspensions, and not ointments. So maybe how the ointment data is treated might be different, and I've not looked into those. Thanks.
I think yes. Just want to hear, for this afternoon we have discussed a lot of like research regarding like brizomycin, and whether FDA has any plan to revise the PSG on this in the near future. Probably for
Yuan, do you mind repeat the question one more time?
This afternoon we have multiple presentations regarding the brinzolamide, and we just want to know, currently this is recommended in vivo, so any like PSG revision for such kind of product in the near future.
Yeah, I think the message was clear. It's an integrated effort really from the in vitro formulation characterization all the way to modeling effort to really help us to de-risk or fill the gaps in terms of what really prevented us from recommending in vitro only approach. For IOP drugs, is more the chronic use, the potential cumulative impact due to minor differences compared to corticosteroids, for instance. Those are more short-term use product. So I think we have made really, as shown today throughout various presentations, we have made significant progress, and I can't give definitive response to this question, but I would say the revision is something as one of the ultimate goals of our research projects, and we are really very close to the endpoint.
Okay, thanks.
Hi, I have another question. My question is for Dr. Vatsala Naageshwaran. So, Dr. Vatsala, thank you so much for your presentation. I think your presentation does illustrate a lot of the challenges that we have in terms of executing these preclinical studies and just potential variability in terms of them. Talking about brinzolamide ophthalmic suspension, as has been quite well discussed today, that particular product does offer quite a substantial challenge from a preclinical perspective if you're evaluating pharmacodynamics because of the intrinsic variability there, but also in terms of ocular pharmacokinetics preclinically. It has a high degree of variability that sometimes make drawing conclusions a little tricky. During your presentation, you mentioned the development of a paired eye study design that could help solve some of these potential issues. I was wondering if you could talk about it and what advantages that method, that approach would have versus regular parallel type of study design, such as what we usually do. Thank you.
Thank you so much. I think the question is from Andre, and we had the privilege of working together on some FDA critical funded programs, including evaluating different formulations of brinzolamide that were developed that were varying in particle size distribution and apparent viscosities that we actually administered in rabbits to look at their ocular pharmacokinetics and look at distribution in different ocular tissues. And when we did the study, we did a statistical evaluation with ANOVA, and it showed no significant differences between the formulations in terms of the Cmax and AUCs. And there were a lot of challenges because of how these critical quality attributes are sort of intertwined also, which I won't go into in this response. But we looked at the sort of as a post hoc analysis, we looked at within animal and total variability, and we identified what was the sample size that was really sort of needed to get statistically significant differences, because to the point made in the question, even though animal studies are very useful, because especially rabbit studies, because they enable us to generate the data that really helps with the validation of the different sort of computational or the PBPK models that can help to predict drug performance in humans. There are some realities about, even with these inbred animals, about the variability we see and the sort of sample size, which from an ethical perspective becomes a problem. Having hundreds of rabbits, is just not possible. It's just not feasible. So what we identified was a design in which is a paired eye design, in which we have the dosing of the reference product in one eye and the test formulation in the other eye, so that we can account for sort of that between animal variability this way, and that really sort of limits the sample size. So we were able to predict what the N number would need to be, and we were looking at just two main tissues. We were looking at iris ciliary body and the aqueous humor, both of which are very important for brinzolamide. Iris ciliary body is the site of action, and aqueous humor is also very important from a bioavailability perspective. So I think that parallel study design versus the sort of paired study design, statistically we showed that the paired design is more likely using a smaller cohort of animals for us to be able to observe statistically significant differences, which can be related back to changes in formulation. So this is the proposed sort of paired eye dosing that, we have ongoing studies to test the formulations in.
Thank you. Just piggyback on the discussion along the line, I think we heard a lot like the similarity between human eyes and rabbit's eyes in terms of anatomy, which is fortunate. So give us, surrogates, right, to really enable certain investigation. So, and today's presentation are really encouraging. But I'm just curious in terms of from your expert standpoint, what are the remaining challenges in terms of cross-species translation and also the extrapolation, the models, for instance, presented today based on 1 or 2 model drugs. So how much of that is also drug specific or formulation specific?
Yeah, so I think one aspect is really having confidence in the permeability of the compounds and how well that's translating across species. I mentioned in my presentation, generally we found that between rabbits and humans, it works pretty well, but there's always still going to be that question with the next drug, So, the question is going to be, can we find something where it is different? And if so, then, we can figure out the reasons why and things like that. Another aspect to consider might potentially be, other preclinical species as well. It's working okay for the rabbit. What about maybe potentially the rat or other animals? I think those would be things that we would want to think about. I think that's probably really kind of one of the major components. Something else to consider as well potentially might be, if there are any compounds that are being locally cleared in the eye as well. That's something that we would generally expect to be very different between species, things like that. So just a few thoughts.
I was talking about this earlier actually with Arto. One of the things that we've run into in trying to model the tear film, which is that all the dynamics there are so important to be able to capture accurate elimination, like you just mentioned. The blinking component of that is very complicated. So we've modeled with evaporation and the blinking for 5 minutes for a rabbit, and if you model that just based on those physics, the rabbit eye will be super dry well before 5 minutes is over. So there's another component or another physics that we're missing. Is it that nictitating membrane that's causing the mixing and hydration of the eye, or what? So I think some of those things that maybe don't exist in the human but do exist in the animal are things we haven't addressed fully yet.
That's a great point. And we've also noticed that taking that blinking rate is going to be really important, especially for things like the ointments. Our model is simpler, so we didn't necessarily need to worry about things like the flow of the tear film. But things like that are definitely important components. I think, like I mentioned in my presentation, again, there's just so many little physiological aspects that you have to take into account. And when you have enough data for one compound to build the model, you can feel pretty confident in it. But again, what about the next model when we don't have as much data?
Yeah, so, right.
Nothing to do, but more and more.
Right. But personally, what I view is uniqueness in terms of generic versus more general application of modeling. If you are given a brand new compound to predict what's happening in vivo, is generic is always comparatively speaking, right? So that's what's really enabled our totality of evidence approach in vitro is we are not almost all cases we are really not necessarily predicting what's happening in vivo. But then what we are really making an assumption is we have very comprehensive, holistic comparison under various conditions for those critical quality attributes, then integrate them together to conclude they will behave similarly in vivo when they are subjected to the same, right? So I think that is a different aspect if we consider only generic perspective. So I guess from that perspective, that modeling approach maybe actually could be simpler, right? You could just, if you could just pinpoint what are really the key, for instance, API properties or certain excipients, API interactions. So I feel like today's talks already give, and I think Mingliang's talk is really encouraging in terms of being able to do the sensitivity analysis. So one goal, Mingliang, you can maybe comment a bit more, is to really use modeling to help us. For instance, for particle size similarity comparison, right now we are recommending population bioequivalence analysis, which we know could be overly stringent. But then we also need data to help us to understand what really is the more clinically relevant space and how then we can modify our statistical acceptance criteria accordingly. So Mingliang, if you want to comment a bit more on that.
Yeah, that's really good point. I think, yeah. So yeah, in generic space, so what we would like to have is to have some like developed and validated model, and then we can utilize that model, to see how the formulation changes or how the formulation in vitro critical quality attribute CQA changes affect the in vivo, exposure mechanistic. That's the ideal scenario. That's the goal we are trying to accomplish it. But during this process, I think the key part is that the baseline model development and the validation part, which I know is like maybe it's product dependent. For some product, maybe we have more data for model development and also validation. For the example, like the brinzolamide, we have relatively like rich data, especially in the preclinical species. Clinical space, for example, in rabbit, we have from literature, we have a lot of data available, both for PK measurement and also IOP, measurement. That's good news. So we started from there. We get some understanding of how the API performed, how they absorbed in the eye tissues. At first, like in the rabbit eye, then distribute, eliminated through eye tissue, so we get some understanding. Then, starting from there, we can translate that to the human models by considering the difference between anatomy and physiology. So that's a good strategy. So in terms of the challenges, I think right now we are facing some challenge, at least for the example we have started, at least I think one thing is that, for the data available for model development and the validation, for example, from literature, there's some like a limited, especially in vivo data available for most products. That's one challenge I would say. Second challenge I would say is that, even there's some literature data available, but there may be some like inter-study variability, large variability, same compound, maybe a same drug applied to, rabbit, similar rabbit, let's say New Zealand white rabbits, but from group to group, the variation is large sometimes. And even within the same group, from different studies, there might be variability as well. So I think that's another, challenge that we needed to make some like scientific decision, what kind of data and how we can utilize those data available, to validate or to develop and validate the model. And then lastly, I think we also, we have some understanding of the, anatomy and the physiology, for example, for brinzolamide in rabbit and the human eyes, but still we need to have a more understanding for something like, for example, melanin, distribution in different eye tissue. So something like that. I think there's some challenges there, but still I think through recent years, effort from all the stakeholders, like the industry, academia, and also government agency, I think we are getting better and better, more understanding. So hopefully one day we can have a really good, cases, something like that, for mechanistically understand this like suspension, for example, ophthalmic suspension, so that we can proceed with that in vitro only approach, or with some in vitro plus modeling approach to demonstrate BE instead of doing some like the clinical endpoint study. Sometimes it's costly, and also for some like in vivo study, of course, for eye tissue, it's almost impractical, and cost prohibitive. But it's for some case, for example, cataract patient, that could be possible, but only small range of the product that might be applied for. But overall, I think we have some direction. Maybe we can work together to make this happen. Thank you.
Thank you, Mingliang. So we only have a couple of minutes left. If we still have online questions, Yuan, you could get ready to ask. But in the meantime, Romit, I want to circle back to see, I don't know if you or your team had a chance to listen to the particle size workshop we held in September, but your presentation highlighting the challenges still, with particle size measurements. I'm just curious to see, from your perspective, do you think that is still something technically challenging? And if that's still the case, then what might be the remaining challenges?
Sure, I think that's a great question. As I mentioned in my presentations, for particle size measurement, ophthalmic suspension is relatively easier compared to ointment particle size measurements, as I explained, and it's getting even more complex when it includes two API. What Dr. Lu mentioned, right? The product, when it comes to two different APIs, there are a lot of complexities where we have not only have to identify A versus B, but we also have to perform the particle size distribution. So those kind of challenges are still there. I know industry is working and trying different ways to resolve those, but I think it's still very complex in terms of proving particle size distribution correct method for those products.
Okay, yeah. And one thing actually we learned through Dr. Lu's and another external project we conducted on ointment is in terms of the particle size. Actually, regardless hydrophilic or hydrophobic, the manufacturing process actually could keep the overall particle size distribution within a fairly narrow range, just because you have to go through a rigorous mixing to really achieve the final content uniformity. Is that something agree with your observation during product development?
Yeah, we have very interesting observation for the particle size. We tried to understand what is the size impact for dexamethasone. When we have two excipients, it's hard to do, so we only did one at one time, right? So we have dexamethasone for the study comparison, and we made a dexamethasone with different sizes. And then we started just look at after preparation, and then you see like the size and different sizes change the release performance or not. The conclusion is there's no difference, no significant difference between different particle size that was introduced before preparation. But even after preparation, we saw that the particle size were smaller, and also that could say that could be one of the reasons you don't see differences. Why the particle size was smaller? So when you're doing the preparation process, you do high-speed mixing, and then you have a hydrophobic ointment with this hydrophobic drug. So the drug could be a little bit more like in a dissolved stage of some of them. So like some of them are smaller because they're dissolved in the hydrophobic ointment base. And that brings you no difference, no significant difference in the end for all these preparations. But based on the literature, actually you see the hydrophilic drugs is purely just the particles, and their particle would not be affected by the manufacturing process without dissolving the ointment. So those particle size matters a lot. So that is correlated to the release performance. So that's our position, again, for distinguishing the hydrophobic versus the hydrophilic drugs.
Thank you. Thank you, Dr. Lu. I think we are running out of time for the virtual portion of today's workshop. Really would like to thank everybody again for the wonderful presentations and productive, insightful discussion. So with that, I will give a very quick closing remark to wrap up our first day virtual. And I really hope everyone has been enjoying this workshop so far. And today we certainly covered a wide range of scientific and regulatory topics, which every single one in a way reminds us, ophthalmic products may be a small, relatively smaller group among all the drugs we have approved, but it has its very inherent complexities, and there are still quite unmet scientific and regulatory gaps. But what really inspired me today is the conversation are evolving. This is our first really solely focused on ophthalmic products workshop, but the workshop we held back in June 2022, 3.5 years ago, we had a portion focusing also on ophthalmic products. Compared to then, I think in the past 3.5 years, through lots of internal, external research efforts, collaborations, we really have brought us to a point where right now we are really talking about true integration. How the formulation in vitro science characterization that connects with in vivo performance, how the comprehensive characterization now are introduced into modeling approach, and how the simulation can maybe bring new approaches for conducting BE. All those can help really bridge the critical gaps to allow more effective or alternative BE or product development strategies. So the shift towards this more holistic, mechanistic understanding of the product is really encouraging. At least personally, that's how I feel. And also I feel like today, our audience, our presenters, panelists are really willing and trying to collectively explore and think differently and test or push for new ideas. Then as we look ahead to our D2, we'll actually turn into another group of complex ophthalmic products, the inserted, implanted drug device combination products, which is relatively new to the family. So those areas also, has its very unique challenges because it blends the drug delivery system, the engineering aspect, the material science, but then they do bring in very fresh challenges, but then also exciting opportunities for us to be innovative here. So with that, I really thank you all for your thoughtful engagement today. I hope the discussion really, you found the discussion really interesting, and for our virtual attendees, enjoy the rest of your day.